Now we switch to three-dimensional representation of proteins. The first statement that should be made here is that the all-atom representations are generally considered to be too crowded or too complex. These are two examples of an all-atom representation. Here, this is a so-called stick representation of a protein, and this is a so-called space field representation. It is not a raised surface because here not the actual electronic distribution is calculated for the structure, but each atom is represented by its van der Waals sphere. So it's close to a surface representation, but it's not an actual surface representation. Still, both of these are too crowded. You can see that there is a mass of different atoms that are colored differently. Here the coloring is by atom type, so the green atoms are the carbon atoms here because the background is black, red is oxygen, and blue is nitrogen, of course. I am talking about an all-atom representation, but it's not quite true, because in these structures the hydrogen atoms are not even shown. It's already very crowded without showing the hydrogen atoms, as many structures determined by X-ray crystallography do not contain the hydrogen atoms as available in the PDB database. This is a quite common representation, all-atom representation, that we don't show the hydrogen atoms. Anyway, they will make the picture even more crowded. On the right, we can see a simplified representation, where in 3D we can see the helices with these cartoon representations, and usually it much better suits our purpose, because here we see the path of the polypeptide chain in the structure. We can see how the fold is generated by the actual chemical structure. Here the coloring is also different. This is a so-called rainbow coloring, where color scale is applied from the amino terminus to the carboxy terminus of the protein. Because all-atom representations are too crowded, we usually turn to specialized representations like the ribbon representation shown before. One relatively often used representation is the so-called C-alpha trace. Here we only show the C-alpha atoms of the protein, and we usually link the C-alpha atoms of neighboring residues with a line. But please note that these are not actual chemical bonds. The C-alpha atoms of neighboring residues, neighboring amino acid residues in a protein, are not covalently bonded. The amide bond is between them. But just to show the path of the polypeptide chain, we use these lines to connect them. You can see that although the secondary structural elements are not explicitly highlighted with ribbons and arrows and cylinders, we can still have some understanding on the helical regions and the extended regions like that are beta strand, that might be beta strands. Here you can see that many structures are shown in this picture. These many structures are superimposed in using this part, this half of the structure, only to show how the other half of the structure is different in the different structures that are shown here. So the purpose of this representation here is to show the extent of diversity of the structures, and because and this C-alpha trace representation was chosen as a suitable way to show that if you superimpose one half of the structure, then the other half of the structures differs a bit, and here you can highlight and depict and understand the differences, the extent of differences, which is not very large here in the structure. So note that this is a mixed representation where the ligands of bind to the protein are shown in an all-atom representation, again with different colors, and it is important because you should see at first that these are not part of the protein chemically, but just bound to the protein with secondary forces usually, and you can see the relationship between the two binding sites, how far these are in the different structures. Cartoon representations are among the most commonly used representations of protein structures. Here the spike protein of the SARS-CoV-2 virus is shown in a cartoon representation. You can see that this cartoon representation here is combined with the linear representation, where the domains, different structural units of this protein, are colored differently along the sequence with these boxes, and this coloring is kept here in the structural representation in order to be able to link the position in the sequence of the given structural elements to the spatial organization in 3D. Okay, so this is some kind of rainbow coloring where we use a color scheme along the sequence, and we keep that color scheme when we depict the structure. You can see that this petal-like organization of this subunit is really clear here. So you can see that we have distinct structural units of this protein, and it can also be used to highlight the position, spatial position of the cleavage sites where the spike protein is cleaved by proteases when it's undergoing maturation. And this is the S2 subunit or S2 part of the spike protein, and this is the S1, which is basically responsible for receptor binding, and this S2 part is responsible for cellular entry. Again, this is another kind of cartoon representation where we use these cylinders instead of the ribbons to depict alpha helices. This protein is again protein of the SARS-CoV-2 virus. This is this RNA polymerase, and the coloring here is used to depict the different functional domains of this protein. This is actually complex because NSP7 and NSP8, which is presenting 2 copies, are also depicted here, but as you can see, there is no RNA in this structure, so this is an RNA-free structure, and this yellow neuron domain is a domain that is specific for the RNA polymerases of the given virus family. Here in the other picture, you can see that this is again a mixed representation where we have a surface representation of some of the domains and cartoon representation of other domains. This can also be useful if you would like to emphasize the spatial relationships and organization of the structures. The names of these domains are come from that related polymerases are usually depicted as similar to a hand or a palm of a human. Okay, so macromolecular surfaces are usually can be used to highlight the diversity of structures. Here in this representation, the diversity of SARS-CoV-2 structures with experimentally determined structures are shown. You can see that that these proteins come in many shapes and sizes and forms, and the coloring here is subunit based coloring, so the different colors highlight that the given protein complex is basically built up from different polypeptide chains, and again we have a linear representation. The full viral genome is depicted here, and this polypeptide which is proteolytically cleaved to give rise to all these proteins is also highlighted here. Here you can see the polymerase again, this time with RNA and with the larger part of the NSP7 and NSP8 proteins, that is also a complex basically of multiple proteins, and proteins with different functions, for example the spike protein and the other proteins are colored differently. So this is a nice view of a zoo. This is more like an illustration than something from which we can which highlights something we would like to understand in more detail, but it's still very useful and spectacular. Surfaces can be used to highlight other or functional features. So here this is the surface of the expressome. The expressome is basically the coupled RNA polymerase and ribosome in bacteria, which are usually or can be in physical contact with each other. And such a representation, this is the polymerase in green, and this is the ribosome. The two subunits are colored with different shades of blue here, and these are proteins that help making the contact between these two proteins. And in this other representation, where this surface is made transparent in order to look inside the structure where other molecules are again depicted with the surface representation, this is the RNA polymerase that binds to DNA and synthesizes the messenger RNA, and here you can see the path of the messenger RNA within this large macromolecular complex to the active site, peptide transfer center of the ribosome, when here are the tRNA molecules. So this helps you understand how the transcription and translation in bacteria are mechanistically coupled. So this is not only they are not only occurring close to each other, they are occurring basically spatially coupled way, because there is direct contact between the RNA polymerase and the ribosome. The bottom depiction shows hemoglobins. The left one is the fetal hemoglobin and the right one is the adult hemoglobin. You can see the coloring emphasis here, that the alpha subunit is common to both of these hemoglobins, but the beta subunit is specific for the adult hemoglobin and the gamma subunit is specific for the fetal hemoglobin. It makes it possible that the fetus inside the womb can get oxygen from the blood of the mother, because the oxygen affinity of the fetal hemoglobin is different from the oxygen affinity of the parental or adult hemoglobin. You can see that the structures of these gamma and beta subunits are basically very similar. So it will be a very nice task to highlight the structural differences. Here the coloring shows the alpha subunit is the same and the other two subunits are different, and you can see the heme group here depicted in red. How can we add more information to macromolecular surfaces? Yes, we can add functionally important information or evolutionarily important information to macromolecular surfaces. This is just demonstration. This particular figure is actually from Wikipedia, but I like it because it's really nice. It highlights the difference between the binding site and the catalytic site in an enzyme. You can see that this is an enzyme. Here we have the substrate with stick representation. This is kind of an all-atom representation. We have the surface representation of the protein, and the residues or the part of the surface that is contributed by the residues in the binding site is colored blue, and the catalytic residues are colored red. And it's clear that the substrate is bound by the blue residues, but the chemistry, the catalysis, is actually performed by the red residues. So it helps you understand why substrate specificity is determined by different residues those that determine the chemical specificity, what kind of reaction will be performed by the enzyme. So this is, I think, a very nice depiction, both in schematic way and both in an actual three-dimensional representation. Another common way to color surfaces is to add electrostatic potential, the charge distribution around the surface, because it's very important in many intermolecular interactions. The charges play important role, so it's customary to calculate the electrostatic potential and put it on a surface representation. You have to use specialized tools for it to calculate the electrostatic potential, and you can put it on the surface with different programs. You also have to put a scale. Okay, so the electrostatic units, I won't go into details here, but what are you actually depicting? What is the scale that you are showing here? And here you can see the differences between these proteins or protein variants where the mutation causes changes, spectacular changes in surface charge distribution. Another picture here shows the residue mutability and conservation. Mutability is what you see in a laboratory when you try to generate mutant of a protein and measure some function, and you assess how the function is affected by the given mutation. And conservation is basically an evolutionary aspect where you gather sequences from many organisms and assess whether a given residue is conserved, is the same, or is easily replaced by other residues in different organisms. And these two aspects are shown in this surface, and this kind of representation very nicely shows you that the mutability and evolutionary conservation give us a similar pattern, but not exactly the same pattern. So you can think of what differences are in an in vitro environment and in an in vivo evolutionary setting that influence the function of a given protein. I would like to say a few words about representations or visualizations that are specific to given structural motifs, especially alpha helixes and related motifs. The so-called helical wheel diagrams can show a helix assumed along its axis, and it emphasizes the residues on the surface and the relative positions of the residues on the surface. Here a so-called amphipathic helix is depicted. You can see that one side of the helix contains hydrophobic residues, and the other face of the helix displays primary hydrophilic residues like lysines and glutamic acid. This is common in some antibacterial or antimicrobial peptides. Helical net representations correspond to representations where you unfold the mantle of the helix, like this, depicted here, and are useful when you would like to highlight the interactions between residues. Here these are specific helixes, so-called single alpha helices, where a regular pattern of oppositely charged residues is characteristic, and these helical net representations are quite useful to highlight the the different kinds of favorable and unfavorable interactions between oppositely and identically changed residues on the helical surface. Cold coils are structures that are formed by two or more alpha helices wrapped around each other in a specific manner, and coil is also often depicted with a modified version of the helical wheel diagrams. Here you can see that the actual coil is does not have a really straight axis, because it's or it has a straight axis, but around this axis the helices are rotating. But in these representations you straight these axes up. That is why you can have 7 residues around one of the wheels or one of the circles. And here this diagram emphasizes the hydrophobic residues in given positions, and that they face each other within the helical interface. These are 7 residue heptad repeats, and the first residue denoted A and the fourth residue denoted D are in the hydrophobic inner part of the coil, at least in a two-stranded coil. This is again another depiction of the simplified depiction of the coil. You can see that the supercoiling is again omitted, and here the ionic interactions between residues in positions G and E are highlighted. Sometimes we would like to compare three-dimensional structures, and this is a topic that is already not only visualization but also analysis topic. So the analysis aspect of this is quite substantial, because without going into much detail in this lecture, I just would like to let you know that protein structures can be aligned to each other in a way that does not consider the sequence. You know that protein sequences can be aligned, and when you align protein sequences, you consider the chemical similarity between the residues. Protein structures can be aligned in a way that you can find the best correspondence between the residues, but in this way you only consider the spatial relationship, spatial organization of the structures, and you ignore whether you align the leucine with the tryptophan or something like that. Okay, so in this, because structure is more conserved than sequence, structure alignment gives you more accurate information when you have to deal with proteins that are evolutionarily related, but their sequence is substantially already different. Here you can see the structure alignments of 2 proteins, and the bottom alignment is a sequence alignment based on sequences alone, and the alignment shown here is basically a sequence representation of the structure alignment. You can see that this is a shorter alignment. Here you can, if you depict the secondary structure elements here, they correspond relatively well to each other in most cases. And here it is ensured that the spatially equivalent positions, residues in the spatially equivalent positions of the 2 molecules are below each other, are aligned below each other, which is not guaranteed by the sequence alignment alone. Okay, so this is quite a topic for a structural bioinformatics class. But if you remember that we can have structure-based alignments, and we can represent these structure-based alignments and visualize these structure-based alignments to highlight the structural similarity, even in the case of low sequence similarity, then it might be useful.